Everybody, this is Miss Clemmy, and now you're going to take a look at muscle contraction and excitation. So to do that, we're going to look at a couple of things in a series of steps, and please sketch this as you go. The first thing we're going to look at is muscle organization. So if we have our muscle here, attached with tendons on two sides, you could say this is like the bicep. And that muscle is divided into lots and lots of smaller parts in bundles after bundles after bundles, but the most important part is the muscle fiber, which is actually the muscle cell. And even the muscle fiber is divided into smaller and smaller bundles, but the one that's really important is the sarcomere. And that's what we're going to draw first. So I'm going to draw a sarcomere, and a sarcomere has two main components. It has a thin filament called actin, and I just drew two sets of actin. And it also has a thick filament called myosin. Oops. We're going to make, and myosin has these weird shaped heads on them. I'll draw one on this side. Oops, wrong way. There we go. There we go. So this is the simplest version of a sarcomere. There's myosin in the middle and actin on the outside. And the whole goal of a sarcomere and muscle contraction is to shorten the contraction. To shorten so that everything's going towards the middle. Now, how does it do that? It does it because myosin naturally wants to attach to actin and swing it forward. And then it'll release, grab another one, and swing it forward. And if you imagine all of these are doing that, they're going to shorten and shorten and shorten and get smaller and smaller and smaller, like a ratchet. Okay? So what I'm going to do now is zoom in on just one portion of the sarcomere to take a look at how muscles contract. So let's erase this guy. All right, we're set. So let's redraw our actin. And I'll redraw my myosin. And just that upper portion. Okay, now, like I said, myosin always wants to be attached to actin, but we know that can't happen because then our muscles would continually be contracted, and that's no good. So we need a blocker. We need a gate that keeps myosin away from actin. So I'm going to draw my gate, or my fence, and that fence is called tropomyosin. Okay. Great. But like any great fence, it has no clue when to let things pass and when to keep things on its proper side. And so we need a gatekeeper. Let's introduce our friend in green. And our friend in green is called troponin. Troponin is the guy that actually controls the gate, when to raise it, when to raise this gate up, and when to lower it back down. But troponin's not just going to release that gate whenever it feels like it. It's going to need a password. And so our password is going to come in the form of calcium. And so once calcium binds to troponin, then troponin is going to simply lift up that gate so that it's up there. And once that gate is up there, then we know the drill. Then we know that myosin can come in and latch on here and swing forward all of those actin molecules. The only other thing that's needed is energy. Is energy in the form of ATP. Now, what's ATP used for? Probably not what you'd think. ATP is actually used to rip myosin off of actin so that it can go back and grab another. Because if you think of the myosin heads as like a locked and loaded ratchet, as soon as it reaches up there, it's going to ratchet forward. It doesn't need energy to do that. What it needs energy to do is to release from that and grab a new one. So as long as ATP is present, calcium's present, we've got ourselves a contraction. Now, everything's fine and dandy, but how does calcium actually know to bind to troponin? Let's erase this and draw up one of our last pictures here. It's of muscle excitation, how it actually gets the signal. So what we're going to draw up here is a neuron. And in that neuron, or nerve cell, we have a chemical. And it kind of slowly moves because your brain is telling you you want to move your hand. So it's going to fuse with the cell membrane here. It's going to release those contents. And as it releases that contents, now here's where it gets interesting. So let's say here's our muscle fiber. Okay, this would be our cell membrane. And for muscle cells, they have weird names for the same things in any other types of cells. So the name for this particular guy, for the cell membrane, is called the sarcolemma. You start to sense a pattern here with all these words having to do with sarco. That's muscles. So the sarcolemma has a little receptor for this little chemical. And that chemical is super important. It's called acetylcholine. So it's the chemical responsible for getting the message to our muscle cells, our muscle fibers. And now what I'm going to draw inside here, I'm going to draw a series of odd-shaped tubes here. And what I just drew is something called the sarcoplasmic reticulum. And it's sitting inside the cell, the muscle fiber, and we give that a special name. It's just sitting in the sarcoplasm, just like the cytoplasm in any other cell. But the role in the sarcoplasmic reticulum, all it does is it stores tons and tons of calcium. It just is chock full of calcium. And so what happens is when acetylcholine gets to the cell, the muscle fiber, that message is going to get propagated to the SR. And as soon as the SR gets that acetylcholine message, it's going to open up its floodgates. It's going to open up its calcium gates, and so calcium can just come running out. And it wants to do this because calcium's going from high to low. Diffusion, passive transport. And so calcium's going into the sarcoplasm, and the sarcoplasm is where all the myosin and actin are found. So it's great. So then it finds the closest troponin to it, and it allows the muscle to contract by attaching onto troponin. Now remember, we also mentioned that in order for muscles to contract, there needs to be ATP. So in this sarcoplasm, there's not only SR, but there's lots and lots of mitochondria pumping out the ATP to also make contraction possible. Now the last piece of the puzzle is this. How do we stop the contraction? Well, we know calcium is involved to contract. So in order to stop, we need to get rid of the calcium. And so what happens is if there's no more signal, if acetylcholine no longer is relaying the message to the SR, the calcium then is going to be pumped back up into the SR to be used again. But now we have to pump it. We have to pump it from low to high. So that actually requires even more ATP. So ATP is used twice in muscle contraction. But now that calcium is safely stored back in the SR, then everything should be back to normal. Then troponin lowers its gate down. There's the tropomyosin is blocking myosin from reaching out and contracting and pushing forward actin. So that's a look at muscle structure and how myosin and actin are used for a muscle contraction. along with acetylcholine, which starts everything from the very beginning. Thanks for listening.